right, so we talked about gene regulation in prokaryotic cells, and of course it's simple. There's a little operon that can be inhibited. As you might imagine, in eukarya, it's way more complicated because eukaryotic cells have a separate nucleus, but also because eukaryotic organisms are often multicellular. So in this, we are going to just sort of go briefly through a bunch of these mechanisms for gene regulation. And I want you to think of it as a plumbing system. And each of these places where genes can be regulated is a valve that can essentially turn the flow of the protein on or off. So we're going to go through all of these mechanisms: chromosome packing, gene transcription, RNA processing, mRNA breakdown and transcription, and polypeptide formation and protein breakdown. Some of this you actually already know. We're going to talk about it a little differently. So let's start out with long-term inactivation of genes, which happens through something that we call chromosome packing. So think of this as the cell taking a gene that it is never going to use and packing that away, just like people in Southern California pack away their snow gear. Okay, you may need it, I don't know if you're going to go skiing, but let's just admit this year night you're not going skiing, so just keep that stuff packed away, right? So a cell in, say, your stomach needs to make hydrochloric acid, but it does not need to make insulin. Cells in your pancreas need to make insulin, but they don't need to make hydrochloric acid. So both of those cells are going to pack away and inactivate whole genes and sometimes whole chromosomes. And when a chromosome is packed away, it's basically coiled up more tightly than it normally would be in the chromatin. So I want to talk about X-chromosome inactivation because it's really easy to understand and it has super cool effects. So X-chromosome inactivation happens very early in embryonic development. So remember that a new organism starts with a zygote, which is a fertilized egg, that then does mitosis a bunch of times and creates a ball of cells called a blastocyst. Within this blastocyst, there's this mass of cells inside that's going to become the new organism. So let's, for this example, say this is going to be a cat. Okay? Well, the cool thing about cats is that the color of the cat's fur is controlled by a gene that's on the X chromosome. So in this pile of cells, if this is a female cat, each of these has 2 X chromosomes, but it doesn't need 2 X chromosomes. You and a cat can survive just fine on the DNA in 1 X chromosome. That's why males that have an X and a Y develop just fine. So one of the X's gets packed away, and it gets packed away at this stage of development. So here we have chromosomes in a nucleus. One of them gets bound to a bunch of other molecules and literally packed away. So the other one is going to be expressed. This is some information from an article that was in Nature a few years ago describing how this happens. So imagine that we have this blastocyst and the X chromosomes, one carries a gene for orange fur and one carries a gene for black fur, or I should say allele. In some of the cells, the orange gets packed away, and in some of the cells, the black chromosome gets packed away. Now, these cells are going to continue to do mitosis and develop into a cat. The spots of the cat that develop from each of these cells are going to have either the orange allele present or the black allele present, and the other one is packed away. As a result of that, the areas of skin produced by the different cells that came from the blastocyst have different colored fur. And so you end up with what we call a tortoiseshell cat that has both black and orange fur in splotches. Those splotches developed from different cells in the blastocyst. Now, why different on the left and right sides? Because in the blastocyst, the certain cells are also determining whether they're going to be on the right or left side of an individual. So it's not uncommon to see an animal, especially cats with this kind of coloring, that have a line right down the middle of their face, one color on one side and one color on the other side. So remember, we talked about different kinds of inheritance in the last lectures, and we talked about fur color being one of the things that was codominant. This is how the codominance happens. Both are present, both the orange allele and the black allele are present, and in each cell, one of the alleles is packed away and the other is expressed. So orange and black are codominant to each other. That's the inheritance. The mechanism of that expression is through X-chromosome inactivation. Okay, does that make sense? So that's a tortoiseshell cat. That's black and orange all over. Now, there's also this little beautiful little kitty. That's my cat. This is a calico cat. So the calico cat has black and orange and white. The white is a gene on a different chromosome that codes for color or no color. If the no color gene or no color allele is activated, then that patch that develops from that gene doesn't have any color, regardless of whether the orange or black allele was packed away when the X chromosome was packed. Okay, so we have two genes working together here. So white, brown, and black are all codominant. Sorry, black and orange are codominant to each other. White is dominant to black and orange, because if white is expressed, if no color is expressed, then it doesn't matter whether the black or orange is expressed. Oh my gosh, so is my cat male or female? My cat is female because she has two X chromosomes, because the black and the orange are on the X chromosome. Make sense? Okay. So here her black allele was chromosome was packed away, and here her orange allele chromosome was packed away. Now, one more interesting take. How do you get a male calico cat? And yes, this is a cat that showed up at a cat rescue or an animal shelter a few years ago, a male calico cat. So how do you get a male calico cat if in order to have black and orange, the cat has to have two X chromosomes, but if in order to be a male, it has to have an X and a Y? Non-disjunction in meiosis. All of it's coming together, all the stuff we learned. So this cat is a male with two X chromosomes and one Y. Yes, non-disjunction on the sex chromosomes in meiosis led to a cat with two X and one Y chromosome. And so X chromosome packing happened because two X chromosomes were present, and so he ended up black and orange. And that, my friends, is how you get a male calico cat. We pulled together meiosis and inheritance and gene expression all in one section. Isn't that cool? In the next one, we're going to pull together what the how RNA processing is going to feed into this process.